Good morning, Hire. Today we are going to look at treatments for infertility in males and females. Have a look at the picture that is presented on the board just now. You should see two people holding a picture of a baby from ultrasound. So I want you to consider what fertility issues might LGBT couples and infertile couples be able to benefit from. So think about it. What kind of things could go wrong in the reproductive system that would help infertile couples, and how could infertility treatments also help LGBT couples? Okay, we will answer these questions through the slides. So infertility in females. There's many different things that can cause infertility in females. Something between 30 to 40% of Scottish couples have fertility problems. It can be due to failure to ovulate, due to blocked oviducts, infections, certain STIs can cause this, fibroids, so non-cancerous growths in the uterus, spasms, stress, and any hormonal imbalance. Infertility in men is usually caused by a low sperm count rather than a lack of semen. So either not enough sperm, so under about 20 million sperm per mil, or an abnormally shaped sperm, or ones that don't move very much. Have a think, so you can pause the video and think what might cause these issues. So quite often it's hormonal problems due to stress, poor diet, or environmental factors like smoking, alcohol, and drugs. So these are some of the things that can be wrong with the sperm physically. This is the normal one, so the acrosomal region. This is called the post-acrosomal region. We then have the tail and the midpiece. These are some of the head defects, some of the neck midpiece defects, and some of the tail defects. So treatments for infertility. You can think, maybe you might know some infertility treatments. So there are fertility drugs which stimulate ovulation. There's artificial insemination. There's intracytoplasmic sperm injection, or ICSI, which is a form of in vitro fertilization. So we can stimulate ovulation. So failure to ovulate can be treated with drugs that mimic FSH and LH. These drugs can have certain side effects like causing superovulation, which is not always very helpful. It stimulates follicular development and ovulation. It gets around the negative feedback effect of the ovarian hormones. That means you can have multiple follicles develop in one cycle. So drugs can be given to prevent the negative feedback of estrogen. So either ones that mimic the action of FSH and LH, or ones that stimulate extra production of FSH and LH. So several follicles will develop. When drugs are stopped, LH is released then, and ovulation will occur in each of these follicles. So this can cause superovulation, which means if the couple are still having sex and the female has produced 6 follicles, which have all ovulated, you could end up with 6 tuplets, which is a lot of kids. This procedure is usually done so that this superovulation will produce enough eggs that can be looked at in IVF. In terms of male infertility, quite often artificial insemination is the answer. So the average sperm count in men is between 10 and 150 million per milliliter of sperm. Semen. Low sperm count can be classified as fewer than 20 million sperm per mil of semen. So artificial insemination is used to treat problems with a low sperm count. So you can see here the sperm in the tube is pushed up through, I don't think the most comfortable syringe in the world. It's not a sharp metal or glass syringe. It is a flexible plastic, but it's still not very comfortable, I don't think. So several samples of semen are collected over a period of time, and obviously these contain the sperm. Artificial insemination is very useful if the male has a low sperm count, because you can take several samples over multiple trips. If a partner is completely sterile, or just there is not enough sperm in the semen, then you can use a donor. So, in terms of IVF, step 1 is the hormonal treatment we talked about before to stimulate development of the follicles. A scan will be carried out to confirm follicles are mature, and the eggs will be removed using a hollow needle. So sperm are collected through the same method as we would for artificial insemination. The sperm and egg are combined in a dish containing nutrient medium. The fertilized eggs are incubated until they get to the 8-cell stage. The healthiest embryos are selected. So if some of the embryos have certain chromosomal abnormalities, there's no particular need to implant those embryos over other embryos that do not have chromosomal abnormalities. More hormonal treatments, such as progesterone, can then be given, and you would scan to check the thickness of the endometrium. The maximum of 2 embryos will probably be transferred into the uterus by implantation, and this raises the question, if you've got 10 perfectly good fertilized embryos, what do you do with the other 8? So this is where we talked about embryonic stem cells in a previous key area. So we can use those for scientific research and treatments. So this diagram is the one that is referenced in your book. It asks you to look at the textbook. This is the figure it's talking about. So if the female has a blocked oviduct, several eggs can be released through the use of FSH, LH, and chemicals that mimic them. A hollow needle is then injected through the suprapubic region to take the eggs from the woman's ovaries. They are put in with sperm in a nutrient medium. Embryos are get to the 8-cell stage. They are checked, and 2 of them would be implanted. We can freeze the other embryos for use in other cycles of IVF. IVF isn't always successful on the first time, and it's usually carried out for the first few cycles under the NHS. After that, you would have to pay to do the cycle. So it's cheaper and quicker, and you don't need to go through superovulation if you store the eggs for the next cycle. So, as we said before the embryo is inserted into the uterus, 1 or 2 cells can be removed and tested for genetic abnormalities. That's why we don't do this at the 2-cell stage, because you can't just take one of the cells. Once it gets 8 cells or more, you can take a cell and test it for chromosomal abnormalities or single gene disorders as well. ICSI, intracytoplasmic sperm injection, ICSI. If mature sperm are defective or very low in number, so if you think, oh, we'll do IVF, and actually the sperm aren't even able to get through the egg when they're in a dish, it's a bit useless. So what you do is you force the head of the sperm into the egg itself using a needle. So the head of the sperm is drawn into a needle and then injected directly into the egg to do the injection. So it's within the cytoplasm the sperm is injected. So ICSI. There's things like ZVF and other ones as well, which you can Google. It's very interesting. But ICSI is the one covered in this course. So we've covered this key area, so we know how ovulation can be stimulated. We should know what artificial insemination is and when it's used, when a donor's semen would be used, and outline the procedure for ICSI. Try and write out the answers to those, and then I'll show the answers. So, ovulation, drugs that mimic the normal action of FSH and LH, and drugs that prevent the negative feedback effect of estrogen on FSH secretion can be used. What is artificial insemination? It's the insertion of semen into the female tract by means other than sexual intercourse. When would donor semen be used? If a partner is sterile, artificial insemination can be used to insert the semen of a donor which has a normal sperm count into the female reproductive tract. ICSI, so the procedure involves drawing the healthy sperm's head into a needle and injecting it directly into the egg. This achieves fertilization in a much easier manner. So hopefully that video made sense. If you've got any questions, please do ask.